Thank you, Munir. Yes, I've known Munir for a few years and can say that he did have dark hair at one point. Thank you to Munir and Christopher and Christine for asking me to give this talk. I started preparing this because I knew to get it through copy approval on BMS I'd need to start early, and therefore I did structure the talk before I got the brief from Vanessa. So I've scrambled backwards to add some new things into this so that we cover some of the things you want me to cover. It was to no avail anyway because copy approval didn't come through from BMS in time, so I'm speaking wearing my integrated medicines hat. So these are my opinions and not those of BMS. So the thing I want to cover is firstly, where did stratification begin? So it didn't begin with cancer, diabetes, etc. We'll talk a bit about infectious disease. Then I'll talk about targeted oncology products, talk a bit about immuno-oncology, and I've already talked to Nick at the coffee break, so I'm forgiven for screwing up BMS's clinical study. And then I'll go on to some future strategies that I think the NHS needs to be aware of, particularly in immuno-oncology. Precision medicine, it's obvious, requires precision diagnosis. We want to get patients into the green boxes, but we don't want to treat them if the drug is toxic and not beneficial, or if the drug is not toxic but is also not beneficial. There's really no reason to be giving people medicines that don't work. So antibiotics, good introduction from Tony around antibiotics and sepsis. This has been going on for years. People culture bacteria, they look and see where there's a clear ring. That indicates that something like New Delhi or ESBL would be sensitive to these additional antibiotics. These strains of bacteria are extremely resistant to many antimicrobials, as you can see. So actually screening antimicrobials and bacteria is becoming quite important. The Neil report came out a year ago now, and it talked about a couple of things that were relevant, and this feeds into what I'd say in the rest of the talk. Firstly, you need to know if it is bacterial in origin before you decide to treat. You ideally like to know what type of bug it is, what's its resistance profile, and therefore what's its likely susceptibility. Now we did a little bit of work looking at the impact of point of care testing. This is something I shoved in, having seen Vanessa's brief. We looked at the impact of point of care testing in A&E when patients were presenting with symptoms of sepsis. And we did it in the following way. One, we actually did primary research in Luton in Dunstable District General Hospital with Dave Kirby, the A&E intensivist. We looked at the patients that went through the hospital in a year to get a baseline indication of what that patient pathway looked like. Then we took some additional information on a point of care test that was available from Thermo Fisher. So we had the performance criteria and the cost of that test, and looked to see what would the impact be of introducing that test into the patient care pathway at A&E. We looked at ICU units, but felt the bigger impact from our research would be in A&E, particularly with a rapid test that was conducted within 10 minutes. So we set some very clear criteria. The impact that we show here is that firstly, the time to treatment is substantially shortened. So you go from 2.5 hours to about an hour to deciding when to treat appropriately. The average cost per patient drops from about 11,000 to 9,500. And mortality rates drop from 22 that we saw at Luton and Dunstable to about 15%. And then in terms of hospital resources, the time spent in intensive care beds dropped from about 8 days to 5.5 days. So this is very much a simulation of the impact of the test. So where we are now is exactly looking at CPRD to get more information, looking at conducting prospective clinical studies to see if we can actually reproduce this impact to give some real evidence that this is where a point of care test might have impact in sepsis. But beyond sepsis and infectious disease, this is a study that was indeed conducted when Munir and I both had dark hair. This is the ADAP study. It came out in 1999. And this is the first example of actually looking at the pathogen genotype to then determine which treatment was given. And you can see that in the control group where there was no genotyping conducted, the drop in viral load was a lot less than it was in patients where you had identified the genotype of the virus and treated accordingly with a set of antiviral agents. So that is really where I think the crucible of stratified medicine lies. Moving on then to oncology and targeted therapies. The important thing to know about targeted therapies is that benefit really only occurs in those that are biomarker positive. So you can see this is a drug, gefetinib, that was developed a number of years ago by AstraZeneca. In patients who are positive, the outcome relative to chemotherapy is very good, whereas those patients that are negative for the mutation, it's like giving them no chemotherapy at all. So it's extremely important in these targeted therapy studies that the patient is identified correctly and robustly. So false positives and false negatives are equally bad in these situations. That said, targeted therapies are extremely good. This is a waterfall plot that shows Xalkori, oh, this is Zelboraf, let me see. Sorry, Zelboraf showing extremely good outcomes in patients. And it was developed by Roche in 5 years from initiating a phase 1 clinical study to approval and launch in the marketplace. And the key thing here was that the regulatory approvals were coordinated for both the drug and the companion diagnostic. So it is possible to develop these targeted therapies extremely quickly. And that's important because the long-term benefits of these therapies are rare. Indeed, they work rapidly, but then you start to see resistance develop and patients soon succumb to disease. And you can see here's looking at crizotinib over 8 weeks, clears a lung tumours. Then at 34 months, you see the tumours returned. When you take a biopsy from that tumour and sequence it, EML-ALK4 has a gene variation. You then put them on serotinib, and then after 15 months, the tumour has further mutated, and you get an additional resistance mutation. So they are extremely effective, but the long-term benefits can be limited. So then on to immuno-oncology. These, as Nick said earlier, this is a very exciting area. We've seen some remarkable advances over the last few years in treating some very, very difficult to treat cancers. One of the things that was recognised early by Hanahan and Weinberg in their initial papers and in the recent review is that tumours seem to resist cytotoxic T-cell killing. That seemed to go against the thinking at the time, where it was known that high mutation burden driving to expression of new proteins and mutations should lead to recognition of new antigens and therefore anti-tumour immuno attack. However, even in tumours that are inflamed, you see no ablation. And this is just an example. I had to put something in from Liverpool here. So this is something from John Gosney showing the types of tumour you see. So you get this immune desert where there's no T-cell infiltration of tumour cells. You get some tumour cells that are restricted to certain areas of the tumour tissue. And then you get widespread infiltration. Even when there's widespread infiltration of T-cells, the tumour still persists. The reason let's see. I'll go on to reasons for that in a moment. So here's just some contrast between targeted therapy and immunotherapy, and why we're so excited about the latter. Targeted therapy does tend to be organ specific. Patients that are negative for the biomarker do not benefit. The benefits are seen early, but the duration is limited, and the biomarker is largely restricted to the tumour cell. Contrast with that, immunotherapy does have an enormous broad tumour potential. Patients still negative for the biomarker may get benefit, and I'll come on to why that may be the case in a moment. The benefit is not always seen early. There is some evidence of what they call pseudo-progression. When you suppress immune tolerance, then you see a rapid influx into tumour of immune cells. That's seen as pseudo-progression. That can lead to early deaths in some cases. We do see extended benefit, and certainly in malignant melanoma, we're allowed seeing durable survival out to 5 years from treatment. There is, of course, that impact in overall survival, and the biomarker is alluded to earlier, is seen both in tumour cells and also in the tumour microenvironment, infiltrating T cells and such like. These products are known as checkpoint inhibitors. There are activating in receptors as well as inhibitory receptors, but most of the products that are in development at the moment are for CTLA4 and PD1 stroke PDL1 in the inhibitory receptors. And there's clear evidence that blocking these inhibitory receptors leads then to tumours being attacked and reduced over time. One of the things that I'm often asked is, anti-PD1, is it the same as anti-PDL1? In many cases, there's an argument for using each and maybe combining these, because as Nick showed, each receptor binds to a different ligand, and so PDL1 binds to 2 ligands, PD1 binds to 2 ligands as well. So, there's potential benefits. However, in some mice models, there does seem to be some differences. Anti-PD1 shows good effect in MC38 mice. Anti-PDL1 shows a lesser effect. So it may be premature to conclude that both approaches are going to be equivalent. So, biomarker positivity in these immuno-oncology products. For genetic biomarkers, it's often a binary outcome. If the biomarker is present, then you'll respond to drug. If the biomarker's not present, you're negative, then you won't respond to drug. With biological proteins such as PDL1, you're actually looking at a continuum. So you're not looking at, is something present or absent. You're looking at how much of it is there. That's got an interesting thing, because when you start to look at these different cut-offs, you're actually changing the relative prevalence of positivity. So for instance, with ALK and EGFR, you're looking at a 5 to 10% prevalence. So as we discussed earlier, you're screening you're screening 20 patients to treat one, screening 10 patients to treat one. With PDL1, it's not quite as bad as that, but where you set the biomarker is quite important. So for instance, if you set the cutoff at 1% and you do it in primary lung tissue, then you're looking at a 70% prevalence of positivity. If you do it in a second line setting where you're looking at archival tissue, you're looking at about a 50% prevalence. When you set the biomarker levels at 50%, then you're actually looking at maybe reducing your patient population that can be treated to about 25% of all comers. So setting that biomarker level is extremely important. And of course, where does that cut-off value relate to response? So does it mean that, for instance, if you're borderline, you're 50%, you're going to be less responsive than someone who's immediately above 50%? It's a huge challenge, and it's something that industry is very much wrestling with, and is actually something that makes it extremely difficult to harmonise testing at the moment. These are some specific problems with PD-L1. So as I said, even patients who seem to be negative for PD-L1 in archival tissue may respond to treatment. So actually, going for that potentially false negative, denying patient a treatment that they may respond to is quite a challenge. The levels of PD-L1 in a patient are quite dynamic. Radiation and chemotherapy are known to variously increase or decrease the level of PD-L1. So when it comes to combination studies, I think sequencing of treatments or combining of treatments in a simultaneous manner is going to be quite a challenge. I've mentioned the dynamics over time. Expression is also heterogeneous. So John and Keith Kerr and Philippe Tanya have all shown me tissue where you can see sections where there's a great deal of PD-L1 staining, and you can see sections where there's next to none. If you start to score one over the other, then you may miss a positive, or you may score something positive that might be negative. So, you know, with all these things, there's noise and error around the threshold that you choose as well. So that makes PD-L1 testing for guiding PD-1 or PD-L1 immunotherapy quite a challenge. Another thought is that pharma's strategies may diverge in some ways. So BMS, they have always favoured an all-comer strategy, because by not setting a PD-L1 cutoff, all patients may be eligible to receive treatment, and it may be that all these patients actually benefit from that treatment. Conversely, Merck, Sharp and Dohn have gone for a cut-off strategy. Initially they were looking at 50%. Recently their cut-offs have been revised to 1% in second-line lung cancer, both in Europe and US. And in the US, they also have a license now to treat first-line lung cancer at that 50% cut-off. The upshot is that testing is required to receive Pembro, but the patient benefits extremely well from that treatment. Although it's fewer patients getting treatment, their outcomes may be substantially better. That is reflected very clearly in the trials that Nick alluded to, Checkmate 26 from BMS, Keynote 24 from Merck. Essentially the same patient population. One of the differences is the PDL cutoff level. With BMS, we looked at a 5% cutoff. With Merck, they looked at a 50% cutoff, and the outcomes are quite stark. Checkmate 26 failed to meet its primary endpoint in progression-free survival. It is no better than standard of care chemotherapy, hazard ratio of 1.15. Conversely, progression-free survival with pembrolizumab is substantially better than standard of care, hazard ratio of 0.5. So here, it's partly we think it's, of course, the cut-off levels, but there's other significant factors we think at play as well around recruitment, study design, etc. It is not down to chemotherapy being better in our study, because I think the chemotherapy curves look pretty similar. And one of the quotes we got at ESMO recently is that a US thought leader said that Pembro versus Nivo really is a tale of precision medicine. Okay. So where is the field of immuno-oncology going? Well, it's heading towards combination therapies. Sharma and Allison put this out as a hypothetical outcome, that we're going to move from where we are now, looking at improved overall survival over a number of years using immune checkpoint blockade. But when you start to combine and sequence these drugs, which becomes a challenge for trial design and trial management, we may start to see more sustained long-term benefit. So this is I think something that we need to work with the CRNs and NIHR to look at how we develop these more complex studies that involve combinations and drug sequencing. So a final thought then towards precision immunotherapies is to say, we're looking at tissue biopsy when either by IC or perhaps by expression analysis, we're looking at the inflamed phenotype versus the non-inflamed phenotype. If you have inflamed, what aspects of it relate to PD-L1, what aspects relate to maybe high gene mutation rates and expression of neoantigen, and therefore can we decide what should be a treatment approach. So for instance, under YES and 1, BMS and Merck are very much looking at PD-1, PD-L1 therapies. Roche is already looking at chemo and IO combinations. Then when you get out to non-inflamed, then BMS and AZ, AstraZeneca, are all already looking at IO combinations, whereas Roche has stated quite clear that it wants to be a leader in combinations using chemo and radio and IO. So the final thought coming back to this point is that we really want to think if the drug combination is more toxic, does the benefit outweigh that increased toxicity? And of course, if the combination is not toxin beneficial, then we should use that. We are winning in cancer, of course, and even high incidence, poor survival, IO products in first and second line are looking very promising. But we are seeing lower incidence but same mortality rates in developing countries now, and this is breast cancer from IARC. One thing that industry is certainly recognising and everyone's recognising is that we need to keep patients very much at the centre of stratified medicine delivery, and I think this is where government regulators, industry, NHS and healthcare providers really need to work in harmony to do that. And this sits very nicely with the final report from the Accelerated Access Review that came out, putting the patient at centre stage of this. And there's lots of stuff out there giving patients advice and the support. And with that, thank you for your attention and happy to take questions.
Thanks very much, Eddie. Time for maybe one question. Nick?
Yes, Nick.
Really insightful. Just what you're saying is suggesting that the development of the biomarker is almost more important than the therapy, that this sort of new type of biological therapy, particularly with these sort of grayscale biomarkers rather than binary genetic. And the dynamic moving target that you mentioned, that the biopsy was taken 2 years ago, is not the same as the tumour you're now targeting.
Yeah.
What are your thoughts about how this is going to be applied in the real world setting of general NHS rather than highly specialist centres that do the trials?
So this is from a personal perspective, not a BMS opinion. There's several ways. So around PD-L1 testing, when we talk to histopathologists, so the interesting thing is that histopathologists, I think, have been almost a forgotten group of stakeholders. They're becoming more important again. If they ever weren't important, they're becoming more important again. So we're actually engaging with histopathologists more from the pharma side, because diagnostic companies really don't talk to histopathologists. They talk to lab techs, they put the platforms in, they give them the kits. So histopathologists were almost neglected. So we are talking to histopathologists, and around PD-L1, they're telling us that it's a very difficult biomarker to read, so it's very important to train them. So we're looking at how do we train histopathologists to read PD-L1. Then, having trained them, we're working with organisations like NIHR, and Merck and AZ and Roche and BMS are all working with NIHR to get an accreditation system so that external quality assurance is maintained as well for that readout. The third bit is thinking about the strategy of how the testing is then conducted in the real world. So having trained pathologists to read this, should you then train every pathologist in the country to read this, or should you go for a number of experts in central areas? In my view is that you go with experts in central areas. So I'm happy to see tests run in a disseminated fashion. So any district hospital can have a platform to run PD-L1 testing. But in terms of reading it, I'm fairly convinced that the reading needs to be centralised. Now, whether that then opens an opportunity for digital pathology, I don't know, but there's a potential need to have that reading centralised. If we go digital, then we take away some of the issues of transferring blocks or transferring tissue. But that's certainly the opinion I would have. So that's a convoluted answer to a simple question, Nick.
Thank you very much, Eddie. So I think one of the points to take forward in the afternoon is that you've described the very complex nature of trials already, but they're going to become more and more complex with combination therapies. And one of the issues is how can the infrastructure that's being developed in the UK make it the best place to undertake those kind of complex trials for pharma as well as for academia in the future.
Absolutely. Yeah.
Great. Thank you very
Thanks,
much.